after exposure, um, B cells, as well as memory cells,
Is this like the third line of defense?
are created. Yes, we're into the third line. Once you start talking about B's and T's, it's definitely third line of defense. And remember that you maintain the levels of these types of cells for quite some time. So maintaining high concentrations of B's and T's provides protection against any future infections. Now, if you think about it, your B cells were all about protecting your body along with the antibodies. Their purpose was to help you out by neutralizing the threat before it became an issue. So your B cells and your antibodies prevent cellular infection. However, you might have an attacker, you might have an evildoer that can make it beyond that point. And so that's when we have to have our T cells come in. Our T cells come into play here. Specifically, the killer T cells. So what they're going to do is they're going to attack and also destroy infected body cells. But I think the question that would exist would be a pretty obvious question. What's the obvious question that would exist at this point? Talking about how these T cells are able to stop or prevent infection, stop an infected cell from continuing on. What's the big issue? You're killing your own cells, and so what does that mean that's so different from the level 2 defense? Remember, like, level 2 defense was recognizing that it was a not you cell. You have one of your cells that's infected. Tell me, what does it look like on the outside? It's normal. So you have to have a mechanism, so there has to be a mechanism. So there must be a mechanism to facilitate recognition of the infected cell. And this system is provided by your MHCs, which I really don't talk much about because they are extremely confusing. But MHCs provide the mechanism for recognizing normal versus unnormal cells. Remember that your MHCs, what that term stands for? Major histocompatibility complexes. And these MHCs, if you will, kind of recognize what's positive or what's negative about the inner workings of the cell. Let's talk about some types of different T cells that are out there. We've established that these T cells are recognizing what is normal versus abnormal cells. So they can take your cells and they can kind of use a little bit of logic, use these MHCs to determine if it's normal or if it's you but not normal. We'll also talk real quickly about the different types of T cells. You've heard of most of these before. There's the helper T cells. You have your killer, or the term bless you, cytotoxic T cells. Then you also have memory T cells. Just as though you had memory B cells, you'd have memory T cells as well. Your helper T cells, and there were no helper B cells, as you recall. These types of cells alerts the entire immune system. Your killer T cells work to attack infected body cells. And your memory T cells, just like your memory B cells, bless you, provide more of a long-term immunity.
So killing T cells and T sub C cells at the
Say that again, I'm sorry.
Killer T cells and T sub C cells.
And cytotoxic T cells?
Yeah, like in the book it just says
Does it just say what?
it just like in the book has it written sometimes T and then sub
Babies.
C.
Oh, like little baby C?
Mm-hmm.
Yeah, so cytotoxic Ts. Let's see. Me?
Where did the natural killer cells
They were back in the second line of defense. They were the cells that just recognize
go?
that
They're not B or T.
they're not Bs or Ts. They're not like an acquired immune system. They're not a learned immune system. This is very specific. You have cytotoxic T cells that kill exclusively one issue or another issue. So it's like you have cytotoxics for the flu 2008, you have cytotoxics for the flu 2012. Your natural killer cells just kill whatever looks abnormal. We'd already talked about the secretion of the enzyme perforin, but just to kind of remind ourselves, what did perforin do? Holes in membrane. And what was the result of that? Apoptosis was the result. And apoptosis, of course, would be basically cell death. Okay. The chapter concludes with like this discussion on like HIV. I think we should be pretty set on understanding like what that is. I think we understand that condition and all kind of the implications of that. Talks about autoimmune diseases. You could know some of this list, like arthritis. Rheumatoid arthritis is like an autoimmune disease. I don't know. Is this the chapter with the picture of the hand? You see that? One of the chapters. There's anyway, rheumatoid arthritis is where like you have issues with your joints, and that's because your cells of the immune system start attacking your body. You could even talk about how some forms of diabetes are autoimmune based. Of course, there's HIV, which is an autoimmune disorder. Allergies, in some degree, are an autoimmune disorder as well. Cool. Is this okay? Okay.
Y'all got anybody call master? Fun have curly hair, fun nails, fun face.
So excretory systems. This is all. This is really is kind of a chapter that's about homeostasis, which again is what this right this major regulation of the internal environment. Right. And when we look at this internal environment, there's a slew of things that we have to be controlling, but I'm kind of focused on some major things that have to be controlled. So I'm going to write down some of the things that I think that you have to control that's pivotal to control, as far as things that are like in the plasma, that are in the water. I would say the temperature of the liquid that's flowing throughout your body. Your blood sugar levels are important to regulate. The amount of free energy, free ATP, free floating in the blood. Water. Nutrient levels. Waste levels. Ion levels. And all of these things, at least a majority of them, maybe not temperature as much, maybe not the ATP as much, but certainly these last several are going to all be able to, with all the, like I guess all the location of all these things is in your blood, so it's really going to be your urinary system, your excretory system, that's going to be having to deal with these pieces, maintaining these pieces. Let's review real quick from the last unit. Circulatory system, what's its major focus? I would say basically they carry stuff. And then your excretory system, its major focus is going to be to gather all the trash from that stuff. So it's basically this massive filtration device. To move out any junk that might be present. So let's spend some time talking about the different waste style products that could be created. I would say the worst thing that could be created is going to be an ammonia based product, or a product that contains ammonia groups in it, as the ammonia is very, very toxic. Somewhat basic. And it's small enough to cross or to diffuse across across a membrane. So you run the risk of having this toxic little guy like in and around your cells, and that's really the last thing that you want. So we have to have a mechanism of removing the nitrogenous waste, and that's kind of what you would put ammonia as being, as a nitrogenous waste. Now, the deal is that ammonia actually isn't produced by our cells. We produce a chemical that's like ammonia. We produce something that's called urea. And what urea is it's a compound that contains these NH groups straddled around a carbonyl carbon. So in a way, at these two locations, you kind of have your ammonia groups. So this right here, this urea, is the waste that is from, or you could see within an animal. Fish actually produce pure ammonia, and birds produce something called uric acid. The uric acid is like what you would classify as bird poop. They go number 1 and number 2 at the same time. That's what that deposit is on your car. Is right? Is their both of their stuff. But your urea is produced by all of your body cells. So this right here is a product of protein catabolism. Is catabolism, is that a buildup or breakdown reaction? Breakdown. So yeah, you break down proteins and you produce this urea, which of course is this waste stuff. So we obviously have to have a mechanism of removing this, and that's actually where your kidneys come in. Now let's make sure that we understand also that this protein catabolism is something that happens everywhere. So that means that this urea is produced throughout all cells of the body. It's produced throughout the entire body. Mostly highly concentrated in the liver, though, where its production is. Mostly in the liver, but can be produced about everywhere. And the idea of this is pretty simple. Running throughout all of your body are these blood vessels. So here's a blood vessel. Say the blood's flowing this way. And inside that blood are a lot of things, some good, some bad. And so I'm going to kind of recognize my little red dots here as being my trash particles, which could be urea molecules. And maybe here with the brown that we see, the brown's going to just signify that it's a good molecule, not a trash molecule. Maybe like water, or an important ion. And as you could agree, it's important that we have a filtration mechanism that takes out the bad stuff and leaves in the good stuff. So there's going to be an organ that facilitates that. It's of course your kidney. And what your kidney does is your kidney accepts in the blood. So there is blood coming into the kidney. And you run it through a variety of filtration devices, but we're going to focus on a filtration device that kind of looks something about like this. It's called a nephron. Nephron stands for the cell of a kidney, just like neuron is to nervous system, nephron is to excretory system. So there you see a nephron, which is embedded in the kidney. Each kidney has millions of nephrons. And what happens is you have a system that maintains the nasty red particles in that nephron, but yanks out, pulls out the good little brown dots that I have. Now what does that mean? The fluid at the very, very end is this good fluid or is this bad fluid? Yeah, this is waste fluid because you have the red dots in there. So here you see waste fluid. Now what do you suspect the outcome of this waste fluid is? Would you keep it?
Yeah.
No, you certainly would not keep it. And what would you do? Well, you would concentrate this down. So you would do your best to concentrate that. Now, how are you going to concentrate something? That's a little hard for us to view, but how would one concentrate anything? Water? Add water, right? Remove water. Remove water. So that means that also, in your nephron, you're working very diligently to remove water. So not only are you filtering out the nasty stuff, but you're able to yank out water as well. And we'll talk about how you start to break some osmotic rules that we've developed maybe as you do that. But as you remove the water, you concentrate the urine more, or you concentrate this filtrate, as it's called, more. And by the time you get it concentrated down, you no longer call it like a filtrate or the blood particles or the blood fluid. You actually would call it, at this point, urine, which of course is going to be dumped off from your kidney to the bladder, correct? So you're going to go through the ureter, land in your bladder. Your bladder has a line called the urethra that runs from it. And the urethra, of course, is the exit point to the outside. So it is the hole by which the urine exits. And it would kind of start to make sense a little bit for in times of lots of water intake, so in times of good hydration, it would be logical, it would appear as though in the case of having tons of water, you don't really have to stress pulling out as much water because you've got so much in your system, so that you don't have to put as much strain on the kidney, so just exit normally. But what if you were in a time of no water intake, or what if you were just bad about drinking your fluids? Well, your body has to work extra hard at this point to pull out any water because you're not taking much in. You'll notice that the urine comes out in more concentrate, a darker color, and you're putting more strain on your kidneys. There's a reason why it's suggested that you have so many glasses of water a day. It's all about protecting your kidneys from excessive strain. And the more unclear stuff you drink, whatever that might be, then the worse off this is. Does that make sense? Now, let's think about when the kidney breaks. So what condition or what do you have to do in the case of a kidney breakage? You're hooked up to a machine. What's that machine called? Dialysis machine? And so in the event that you have a dialysis machine, what you do is you basically pour it into your blood vessels. You hook your body up to a dialysis. I think that's how you spell it. Dialysis machine. And what the dialysis machine is going to do is it's going to filter out your blood just like a kidney would do. The problem is that you have to hook up your body to this machine every couple of days to clear out the nasties. At least every 2 or so days, maybe 3 at best. In the event that you don't, and you would be using a dialysis machine in the event that your kidneys had failed, you had gone into kidney failure, this dialysis machine is going to just yank out all the nasty stuff. And in the event that you don't, or your dialysis machine has or you don't hook yourself up to one, and you do need one, your blood becomes full of trash, basically, and your system shuts down from the poisons that you've developed, that you've accumulated along the way. So it would make sense that you would need a system that is able to pull out all of this waste. Cool? Does that make sense? Okay. For tomorrow, what we're going to talk about tomorrow is we're going to talk tons about the mammalian kidney and how its structure is. I would say your focus for tomorrow is going to be on studying the structure of the kidney and then all of its little parts. So that's your